We're going to take a look at the regional specialization of the renal tubules. So different parts of the renal tubules have different jobs. The proximal convoluted tubule is where the majority of reabsorption occurs. 65% of the filtrate volume is reabsorbed there. It is non-regulated, meaning that hormones do not control the reabsorption in the proximal convoluted tubule. The loop of Henle in the juxtamedullary nephrons is very important because it's going to make the medullary osmotic gradient. And that will allow you to reabsorb more water and concentrate your urine when the filtrate gets to the distal convoluted tubule and collecting duct. And then reabsorption and secretion in the distal convoluted tubule and collecting duct is regulated by hormones. So here's just a summary of the characteristics of the renal tubules. I would star this. This is really important to know. The proximal convoluted tubule is highly permeable to water and solutes, and it's not regulated by any hormones at all. In the loop of Henle, the descending limb that's going down into the renal medulla is permeable to water but not solutes. The ascending limb coming back up towards the renal cortex is permeable to solutes but not water. So they're just the opposite of each other. The distal convoluted tubule and collecting duct are permeable to water and solute, but they're going to be regulated by hormones. We're going to focus on the proximal convoluted tubule. So the proximal convoluted tubule is called the mass reabsorber, and that's because 65% of the filtrate volume is reabsorbed here into the peritubular capillaries. So 65% of the water and sodium is reabsorbed, and then 100% of the glucose and other nutrients like amino acids and water-soluble vitamins are going to be reabsorbed here. This is non-regulated, so there's no hormones that are going to regulate the proximal convoluted tubule. The epithelial cells that make up the wall of the proximal convoluted tubule, the apical membrane has microvilli. So this is the side that's pointing to the lumen where the filtrate is. The microvilli form the brush border. The reason that the brush border is there is that it increases the surface area, which means you can absorb more substances. There are tight junctions between the epithelial cells, but these are unusual tight junctions because they're leaky. So things can move between the cells through those tight junctions. Sodium reabsorption is the key to reabsorbing the rest of these substances in the proximal convoluted tubule. So I would star this. This is really important to remember that sodium reabsorption is the key driving force that allows the reabsorption of the other substances. So in order to reabsorb sodium from the filtrate of the urine and put that back into the blood in the peritubular capillaries, on the basolateral membrane of the proximal convoluted tubular cells, we have sodium-potassium pumps. And then on the apical membrane, we have secondary active transport using sodium. So I would star that and know that that's pretty important because sodium is so key to reabsorbing the other substances as well. We're going to start drawing this crazy picture of a juxtamedullary nephron. So in the renal cortex, I have shown here the renal corpuscle. So we've got the afferent arteriole coming into the glomerulus. Here's our efferent arteriole exiting the glomerulus, and then that goes into the peritubular capillaries. The filtrate that's filtered out at the glomerulus enters Bowman's capsule and then starts going down the proximal convoluted tubule. I also have drawn here the juxtaglomerular apparatus. You can see the granular cells wrapped around the afferent arteriole. Remember, they're going to release renin, which will help increase the blood pressure. And then here's the distal convoluted tubule showing the macula densa cells. And remember, they sense the sodium chloride that's in the filtrate, and they will help control the glomerular filtration rate. Here we have the epithelial cells that make up the wall of the proximal convoluted tubule. And you can see the microvilli, which form the brush border. And remember, that increases the surface area so you can reabsorb more things. So remember, the big job of the proximal convoluted tubule, it's the mass reabsorber. We're going to reabsorb 65% of the filtrate volume here. So we'll reabsorb 65% of the water and 100% of the valuable things like glucose, amino acids, and some of the vitamins. So let's just take a look at sodium reabsorption first, because remember, reabsorbing sodium is going to be the driving force to reabsorb all the other substances. And remember, with reabsorption, we're going to be taking the substances that are in the filtrate and then reabsorbing them and putting them back into the blood. So on the basolateral membrane of the proximal convoluted tubule, we have our sodium-potassium pump. So remember, we're going to be pumping 3 sodium out and 2 potassium into the cell. And remember, this is primary active transport. So we're directly using ATP. I didn't draw the ATP in here just because I didn't have a lot of room. So by pumping the sodium out of the cell, we're going to keep the sodium low inside the cell. So that allows sodium to move from the filtrate, where we have a lot of it, into the subepithelial cell, and we're just going to go down the gradient there. And then glucose is going to go up the gradient because there's actually more glucose inside this cell as it builds up as we reabsorb it. So sodium going downhill and glucose going up the gradient, this is secondary active transport. So we've got primary active transport of sodium on the basolateral membrane, and then we're going to be using secondary active transport on the apical membrane. So by reabsorbing sodium, moving the sodium from the filtrate into the blood, that's going to drive the reabsorption of all of the other substances. So one of those is glucose, and glucose is very valuable. We usually reabsorb 100% of the glucose. Unless you're a diabetic who's not controlled very well, then you end up with too much glucose in the urine and the carrier proteins get saturated and we can't reabsorb it all. And so some of it stays in the urine. But we're going to be moving glucose into the cell by secondary active transport on the apical membrane. And then on the basolateral membrane, we're going to be using facilitated diffusion with a carrier protein. And then we can just take that glucose and put it into the blood and save that so we don't urinate that out. Amino acids and water-soluble vitamins, which are the B and C vitamins, they will also be transported in the same way as glucose. So they'll do secondary active transport on the apical membrane, and on the basolateral membrane, they'll do facilitated diffusion. So we also have water that will follow the solutes, because as you're reabsorbing all of that sodium, water is going to follow that. Some of the water will do the paracellular transport going through those leaky tight junctions, and some of them will go through some aquaporins as well. So we're going to reabsorb 65% of the water here and put that back in the blood. And because we are reabsorbing sodium from the filtrate and putting that back in the blood, chloride ions like to do whatever the sodium ions do. So if we're reabsorbing sodium, we're also going to be reabsorbing chloride ions. We're also going to be reabsorbing potassium ions from the urine and putting that back in the blood. And then the other thing that we can do at the proximal convoluted tubule is we can help regulate the acid-base balance. So we can secrete hydrogen ions. So we end up from the blood, we have too many hydrogen ions, we can secrete them into the urine and urinate those out and get rid of them to help regulate our acid-base balance. And we can also reabsorb bicarbonate ions, which will also help increase the pH and make it less acidic. So this is just pictures from your book. I just had everything in one spot, but this is just the same thing again. Just remember that sodium reabsorption is the key driving force for reabsorbing everything else in the proximal convoluted tubule. So we've got the sodium-potassium pump on the basolateral membrane, and then we're going to use secondary active transport on the apical membrane. And then this one's also showing you the chloride ions. So if you're reabsorbing sodium, chloride ions will follow the sodium. And this is just showing the reabsorption of glucose. So I would star this, make sure you know this. So on the apical membrane, we're going to reabsorb the glucose through secondary active transport. So sodium will go down the gradient and glucose will go up its gradient. And then on the basolateral membrane, we'll do facilitated diffusion. And remember, that's passive. We're going to go from high to low concentration and reabsorb the glucose. And then as we talked about before, 100% of the glucose will be reabsorbed unless you've got too much glucose in the blood, like you're diabetic. Then you can end up with glucoseuria because you saturate those carrier proteins and you reach that renal threshold. And then we're just going to reabsorb the amino acids and water-soluble vitamins, which are the B vitamins and vitamin C. They're going to be reabsorbed just like glucose is. And this is just showing you the reabsorption of potassium. So some of it does go through the leaky tight junctions. And then the potassium that we're pumping into the cell through the sodium-potassium pump will leak back out through leak channels. And then we can also do some transport across the apical membrane too. And then just remember that water is going to be reabsorbed by osmosis. And we're going to reabsorb 65% of the water here in the proximal convoluted tubule. So water is just going to follow the solutes. So as you reabsorb all these solutes back into the blood, water will just follow the solutes. And then we can regulate acid-base balance in our proximal convoluted tubule. We can secrete hydrogen ions, put it back into the urine if we have too many, and reabsorb some bicarbonate ions, and that'll help increase the pH back up to normal if we're too acidic. You don't need to memorize any of this. It's pretty complicated, but just realize that you can do some acid-base balance here, secreting hydrogen ions and reabsorbing bicarbonate ions. And then just a summary of the proximal convoluted tubule. The reabsorption of sodium is going to drive the reabsorption of all the other solutes and water. And remember, the proximal convoluted tubule is our mass reabsorber. So 65% of the filtrate volume will be reabsorbed here, so 65% of the water, and then 100% of the valuable things like glucose, amino acids, and the B and C vitamins. We can also regulate our acid-base balance here by secreting hydrogen ions, so taking them from the blood, putting them into the urine, and then we can also reabsorb some bicarbonate ions. The other thing to remember, I forgot to point this out on the picture I drew, that the filtrate, the urine, and the blood are isoosmotic. So they're going to have the same osmolarity. And we'll see that change when we get to the loop of Henle.